Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host. The real power of gene editing is being able to choose exactly where to edit a genetic disorder, as easily as correcting a tiny typo in a text. With its 25 years of expertise, Cellectis was the first company to translate gene editing tools into potentially life-saving therapies. They invented an allogeneic approach, which is the transfer of cells from one individual to another. And Cellectis' technology, TALEN, can make very precise edits with limited to no off-target effects. And we're learning more about TALEN, this gene editing technology, in this episode. And so that I can learn about it along with you guys, I'm joined by Dr. Julien Valton, who is the Vice President of Gene Therapy at Cellectis. So thank you so much for coming on. I was telling you before we hit record that I have wanted to do an episode about TALEN for a long time because listeners know I'm really enamored by gene editing. And I cannot believe we haven't had an episode about TALEN until now. So thank you so much, Dr. Valton, for coming on and just diving into all this with me.
Well, thank you for the invite. Happy to be here.
So before we get into this very exciting gene editing technology called TALEN, I wanted to talk a little bit about kind of the background in terms of Cellectis. You guys are celebrating 25 years. It's your 25th year anniversary this year. Could you provide an overview? I mean, obviously we can't talk about 25 years worth of content in one episode, but what has been some highlights from Cellectis over the last, few decades?
Well, Highlights was the original idea in 1995 by David Sourdive and André Choulica. They had this crazy idea of using those tools, to edit the genome in a very specific manner. And it became real, like in the last, I would say 15 to 20 years ago. They invented the meganucleases. They started with the dinosaur of the CRISPR and TALEN called the meganucleases that needed a lot of edits and a lot of mutagenesis to make it specific to a given locus, to edit a locus. And then we moved on to the TALEN, which was originally developed by our CSO of plant science, Dan Voitas. And we moved on to the optimization of this tool, the assembly of this tool, and we in 2012, I think, decided to use this tool to produce and develop this new next generation of CAR T cell therapy, which you called allogeneic CAR T cell therapy. And since then, we evolved and used it in clinic, and we'll talk about it, I think. But
Yeah.
this been a great journey so far, and very exciting for everyone in the company, I would say.
Yeah. And I think in the last, year or so, people in general, not just in our field, but the general public is so much more aware of gene editing than they were before. There's just been a lot of major updates in our field and, affecting patient care directly now, which is very exciting. So you've mentioned TALEN, and I'm really excited. So we have to just start talking about it. So for people that don't know, what does TALEN stand for, and how does it work? Because it's, we're going to be kind of comparing it to CRISPR in some ways, because I think people, if they've heard of any gene editing, it may be CRISPR. So I really want to talk about how TALEN works in this episode. So what does it stand for? How does it work?
So TALEN stands for transcription activator lag effector. So it's a very crazy name that originates from TALs, that are transcription factors that were originally, I would say, identifying plant pathogens, and that contain a exquisitely specific DNA binding domain and a transcription activator domain. So what we've been doing actually is to recover this DNA binding domain and then graft it into a nuclease domain. And therefore, a TALEN is the nucleus version of the TAL, and it works by pair. So it has to dimerize on a DNA sequence on a left and right binding sites. The dimerization brings the two nucleases together that needs to dimerize in order to catalyze the double strand break. And so this activity is highly specific because it needs two binding sites to recombine the nuclease domain and cut the site of interest. And it is also super precise. Why? Because we do not have any hindrance of designing TALEN. We just need one T at the beginning of the targets, at the 5' end of the targets. And if you had that, we can basically design a TALEN. So very precise tool because we can design a TALEN every 7 base pair within the genome. So you can imagine the amount of TALEN that we can design per gene, per intron, per exons, per regulatory elements. And so I would say it's a pretty mature tool so far because it's been one of the first one to be implemented in clinical trials. We've done our first compassionate clinical trials in 2015, treated the little Layla with this allogeneic CAR T cell. So I would say it's pretty mature so far with respect to other tools that are coming into the space and are very competitive as well.
Yeah. So I think the big part that is so interesting to me is how precise it is. I mean, you said every 7 base pairs ish, you can target that area. And I would think that because you are targeting the beginning and the end, if I'm getting that right, it's even more precise than, say, other types of gene editing because you have both of those sides. So it's a little bit more confidence in terms of, are we getting exactly where we want to be and not having as many off-target effects?
Yeah. So there is two things here. I'm talking about precision, but also specificity. Precision is more about where you want to target your gene editing tool and if you are able to do so. With the TALEN, you are able to do so every 7 base pair. For CRISPR, for example, you need a PAM domain, right? A PAM sequence that is a 3 base pair sequence, NGG, that is required to be able to design a CRISPR. Here, we just need a T at the beginning, at the end of the targets in order to design the TALEN. That's what I'm talking about, precision. Now, specificity-wise, if we talk about that, we need to talk about the length of the target that needs to be recognized by the TALEN. For the TALEN is 2 times 16 base pair, and those 2 times 16 base pair needs to be adjacent to each other so that the TALEN recombine and cleave. So it's a total of 48 base pair that needs to be here to be recognized and cleave. If you think about it, the probability of finding somewhere else a 40 base pair target is pretty low compared to 20 base pair targets that is required for CRISPR design, for example. So in other words, the TALEN, because of the length of the target it needs to recognize, it's becoming pretty specific with respect to other tools. Make
Yeah.
sense?
So, yeah, very much so. It's almost like when you go to search a document, if you search the letter E, it's going to come up so many times, right? But if
Exactly.
you search a full sentence, it's probably only going to come up once. Is that a fair analogy?
That's a pretty good analogy. Thank you very much for this one. It's the first time I hear that, but that's pretty good.
I get bonus points for that, coming up with I don't know if it's quite original, but so, and I know in the beginning you mentioned CAR T cells.
Yeah.
Can you kind of expand a little bit more on what the role of those are in gene editing, and if it's anything that you were mentioning that maybe I'm missing?
So CAR T cells. So as the people that are listening to this podcast know pretty well, so CAR T cells are T cells that are engineered to recognize cancer. Originally designed in early 2000, they originate from patient T cells that are extracting from the body of the patient. They are engineered ex vivo in order to be trained to recognize cancer and then before being re-injected back into the patient. So it's an autologous CAR T cell therapy, one patient, one drug, one treatment. Now, it's pretty efficient. It's really efficient. It was actually a revolution, and I guess it's going to be the next Nobel Prize, right? But the thing is, it's a $1 million or $2 million therapy, right? So when the first couple of papers were published in 2012 by Michel Salin and Carl June separately. We reasoned that we could make this autologous CAR T cell therapy democratized to everyone, actually. And in order to do so, what we design is a universal CAR T cell that, instead of coming from a T cell from a patient, one drug, one process, we recover T cell from healthy donor available off the shelf that we re-engineered so that it re-express the tumor recognizing domain, which is named the CAR, chimeric antigen receptor. And in addition to that, we use our gene editing techniques to render the cells invisible to the immune patient of every single kind of patient, so that this CAR T cells is universal and can be used for any kind of patient as an off-the-shelf product. And this is where the gene editing, the TALEN gene editing was leveraged to render them invisible to the immune system of the patient that we treated. So that's the basis of the involvement of the gene editing in the CAR T therapeutic space.
Yeah, that's so interesting. And yeah, a huge part of that is if you're giving someone cells from a donor and it's not from themselves, it's not like a self donation, then yeah, the first thing I was thinking about as you were starting to talk about this was, well, aren't they going to have an immune reaction? Be like, hey, your immune system's like, this is foreign material. We got to get rid of this. But being able to kind of cloak it in this invisibility cloak, and so that the immune system isn't fighting it and allowing those cells to basically fight cancer in terms of, you know, its use there. And is there anything that needs to be personalized when giving it to patients, or it really is just, as you said, kind of off the shelf?
Exactly. That's the main idea of what we developed so far in the last 15 years. It has to be off the shelf, readily available. If the patient is diagnosed, it gets a treatment from his oncologist right away. He doesn't have to wait for 13 days for the manufacturing process to occur. It has the product right away injected. And thus there is a percentage of attrition that is much lower than autologous gene therapy. This is one advantage among others of using universal CAR T cell compared to autologous.
Right. And just, you mentioned the cost of treatment like this when it is more personalized and that using that precision medicine, that's so expensive, $1 million, $2 million. Whereas if you're taking something off the shelf, I got to imagine that's going to lower the price, and then that allows more people to be able to use this.
I would say it's more like allow more people to use it as readily available products. Now, I'm not a business people. I have no idea about the price, but I guess that the cost of good is lower. But the efficacy is on par with autologous CAR T cell therapy so far, with the result of the clinical trials that we got up to now.
Wow, that's remarkable. And from what I'm kind of learning, you know, Cellectis has been a leader in this space. Is the TALEN technology intellectual property of Cellectis, or do other companies also are developing TALEN? How does that work?
Yeah. So when we originally developed this technology, we bet on it because before the CRISPR revolution, there was the TALEN revolution. This tool was amazing. It's still amazing. And we sensed that in 2010. And we acquired a couple of different licenses. And on the top of that, we built a very strong portfolio around the TALEN. So the assembly process, the optimization of the precision, the specificity. We have basically 15 years of experience in using the TALEN, optimizing the TALEN, and using it for gene and cell therapy applications. So that was a bet, and I think that was a great bet at that time. And so far, we have a very strong and non-diluted portfolio of IP around TALEN and TALEN application.
Wow, very cool. So it's certainly more complex than just having all TALEN technology under your umbrella, but within that, there are certain patents and different things within the TALEN technology that
Yeah.
Cellectis has and is working on
Yes.
exclusively.
Yeah, yeah.
Interesting.
So it's not only on the tool, but it's more also on the application. Now we are densifying our portfolio on the application. So synthetic biology on T cells, engineering a lot of different cell types. So T cell, B cells, HSPCs, hematopoietic stem and progenitor cells. I think we're going to talk about it today. And it's more about like engineering. Today, it's engineering smart cells that are able to answer a given therapeutic problem. And we've done that for T cells. We're addressing hematological malignancy, but also solid tumor. And this needs additional edits for sure in order to make sure the CAR T cells can understand the cues, the environmental cues that is super hostile for it. Solid tumor has evolved in order to be super hostile for the immune system. I'm sure you know that. And we need to engineer different genetic circuits within the T cells, within the CAR T cells, so that it finds its way towards the cancer without being inhibited and so on. So TALEN, I think, or gene editing as a grand scheme of things is very key here in order to advance the cell and gene therapy field, I would say.
Very much so. And I wanted to talk about one of the papers that you're recently a co-author on about using technology, the TALEN technology, to edit hemopoietic stem cells and progenitor cells. The link is in the show notes for people that want to check out the article, the journal that you wrote. What is the purpose and the goal of this study, to kind of just give an overview before we dive into all the details of it?
Yeah, sure. Well, so this is an approach that leverages the TALEN technology to edit HSPCs so that they reach the brain and secrete a therapeutic protein that is missing in MPS1 patients. So in order to do so, we recover HSPCs and we insert these therapeutic genes, and we know and we use the natural capacity of HSPCs to differentiate into macrophages or myeloid cells, I would say. And those myeloid cells have a natural capacity to cross the blood-brain barrier. And in other words, we use those myeloid edited cells as a Trojan horse to cross the blood-brain barrier and secrete, more specifically in the CNS, the therapeutic protein of interest that is missing once again in the MPS1 patient. So that's the basic idea of the paper.
So when approaching this study, what led to the decision to use MPS1 as the condition that you're focused on?
Well, MPS1 is a rare disease, devastating diseases that affects young pediatric patients, but also like young adults. And the only therapeutic option for those people is enzyme replacement. So protein of interest is infused into those patients in order to rescue the missing enzyme in those patients. This therapy is efficient, but not optimal, I would say. It requires the patient to go into the hospital every other week. It triggers some immune response. And last but not least, the enzyme cannot cross the blood-brain barrier in an optimal fashion. So we thought that in addition to be a very important rare disease to treat, there was also a very good example or like a very good option to establish this proof concept of using the HSCs as a Trojan horse to vectorize a therapeutic protein within the CNS.
Wow. So it's really impressive that you are able to get through the blood-brain barrier by using this. So how does your new approach, from what I'm gathering, it's called promoterless intronic gene editing approach. How does it differ from the usual methods? Is it this approach that allows to get past the blood-brain barrier? Is it kind of two different concepts that I'm combining.
Yeah, so I have to say something. Uh, the ability of HSC to differentiate into macrophages and the ability of macrophage to cross the blood-brain barrier is not new. We know that from like pretty long ago. Now, what is new is in this paper is that we didn't want to have a systemic secretion of the therapeutic protein within the body. We know that HSCs, when they differentiate into a whole lot of different blood cells. And we didn't want all those hematopoietic cells to secrete the therapeutic protein. We only wanted the macrophages to be, or the myeloid cells to be able to cross the blood-brain barrier and secrete the therapeutic protein. Now, how did we do that? We rewire a myeloid-specific gene in order to secrete the therapeutic protein of interest in a myeloid lineage-specific manner. How did we do that? We use our TALEN technology. We targeted an intron of the CD11B gene, which is a myeloid-specific gene, and we inserted a promoterless cassette encoding the IDOA therapeutic protein. When we do that, we totally rewire the regulatory element of CD11B in a seamless manner so that IDOA, the therapeutic protein, can be expressed just as well as CD11B. CD11B being a myeloid-specific gene, IDOA will be and is actually secreted in a myeloid-specific manner.
Wow. And I'm just thinking with this new approach, just how much, obviously we've talked about this study focus on MPS type 1, but how this could be applied to so many other metabolic and neurological disorders. Is that something that your team has talked about in terms of, okay, this went well in this study, what are the next steps to expand this?
Yeah, I would say that's the beauty of gene editing and DNA vectorization into the nucleus. So the therapeutic protein, the therapeutic gene that we're using can be easily swapped out for another one. We now know what are the missing enzyme in the different LSDs, so lysosomal storage disease, and we can easily swap out IDOA for ARSA, which is missing in another LSD, or for GAL-C, or etc., etc. So that methods of intron editing is transposable to any kind of LSD diseases. And I would say more, we can use these methods of myeloid-specific expression of a therapeutic protein for other like neurological diseases such as Alzheimer's or you name it. So it's expandable, I would say.
It's like you've created this process where you're like, okay, this is how it works. Insert what you need in terms of that gene. What does that gene then, go into making this enzyme that there isn't enough of? There's an insufficient amount, and creating this process is just remarkable because of just how much you just mentioned a bunch of different disorders where this could be really helpful. Is there one that you think of in terms of, well, this would be a really good setup to be the next condition to focus on for the next study? I'm pressing a little bit because I don't know if I'm allowed to ask. You can also say, it's a secret.
Well, if you ask me, I'm from innovation. I want to address everything.
Yes.
So then there
Good
is
political answer.
No. I'm not joking. Like we can, and we did that in our paper. Like we started addressing that by expressing other protein. But what I want to say is that this system is not only able to address different disease by secreting in a myeloid-specific manner a therapeutic protein, but it can also address other disease that requires not myeloid cells, but lymphoid cells to re-express in a specific manner a therapeutic protein. We can rewire not myeloid cells, but also like CD4, CD8, I don't know, all the genes that you can imagine to make it lineage specific with a different tropism, I would say.
Wow, that expands it even further. You're like, we can look at different cell lines and say, okay, what do we need to be, tweaking to be able
Exactly.
to alleviate symptoms? So with this, is it more of a prevention of if we have a diagnosis, and especially, a lot of the conditions we've mentioned do affect kiddos. So is it important, and I know a lot of this is still in clinical trials and being studied, but eventually, when this is going to be FDA approved and out there for clinical use, is it going to be really key to have this early on with symptoms or even pre-symptomatic? Or can you have someone that has dealt with symptoms for years and reverse some of those? Because there's a lot of different treatments that it's only in the prevention space. I feel like it's very hard to reverse
Yeah.
damage that's been done.
I mean, pre-symptomatic would be better. I would say it's always better. Now it's a balance between like the benefits and the risk associated to this kind of gene therapy. And I would say like we will be able to answer that in the near future once we have like further data at the preclinical level, but also at the clinical level.
Yeah, we're going to need more clinical trials and everything to explore all this. And one thing I wanted to talk about before we ended is that Cellectis has recently entered into a partnership agreement with AstraZeneca. Can you tell us about this collaboration? Are there any specific diseases you're hoping to target with TALEN in the near future with this partnership?
Sure. Yeah, we're very proud about this new collaboration with AstraZeneca that is very fruitful, I would say. Well, the idea is that we made this deal on combining the power of AstraZeneca with our know-how and expertise in gene editing and cell therapy in order to address immuno-oncology therapeutics, or to develop new, I think, 10 different immuno-oncology products, and also for rare disease. And I guess what I can say about that, it's a very powerful combination of two different expertise. On one side, our cell and gene therapy expertise and gene editing expertise. Um, you can imagine the whole process development of cell manufacturing that we put together in Paris, in New York, and in Raleigh. With all the team that were involved, we have a very strong knowledge in this. And I think this will be complementary with AstraZeneca knowledge in the immuno-oncology field as well. And I think we hope for the best, but I think that so far it's going pretty great.
Yeah, that is just wonderful. I'm just so excited to hear how all of this progresses. And we really are in the age of, genetic editing. It's very exciting to see how far things have come. Something that if, people listen to many episodes of DNA Today, they know that I mentioned, we launched the show in 2012 when that big CRISPR paper was published. And so it's just been so cool the last 12 years as our show has, gone on to see, well, how has gene editing changed over the last 12 years? And Cellectis was in the game even before that. I think you said 95.
Yeah.
So it's just really exciting to see how much progress is being made just within my lifetime. And just in the lifetime of this show. So Dr. Valton, thank you so much. I mean, we covered so much material. I mean, we talked about, so much like in terms of like the immune system and how that works in terms of treating cancer. We're talking about the metabolic and neurological conditions that we could be targeting with this, and really just the system and the process that you guys have been fine tuning to be able to say this is working. And now we can apply this to other conditions as all of these clinical trials and more studies are happening. So I think we got to have you back on when there's the next aspect of when things are, nearing the end of clinical trials, and kind of just seeing how that's going. But this has just been very cool. I was looking forward to this episode and you certainly delivered. So thank you so much.
Thank you. Thank you for your time.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in the world of genetics.